Our next presentation for this morning's session is entitled Physiological Quality Gain in Peanut Seed During Development, and our presenter is Leticia Moreno.
Thanks for the introduction, Dr. Cameron. I will share my presentation now. Okay, so as Dr. Cameron said before, my name is Leticia Moreno. I am a PhD student at the University of Georgia, and today I will talk about physiological quality gain in peanut seeds during development. So the use of seeds with high quality can result in several benefits for crop productions, and examples of these benefits are fast and uniform emergence of seeds, less disease and insect problems, can also increase yield and help seed producers to maintain seed quality until harvest time. So seed quality is a combination of components that together will describe the potential performance of this seed lot. And physiological quality is one of these components. However, physiological quality is not just related with the germination, or in other words, the radicle ability to emerge, or related to vigor, whereas the potential of these seeds during germination and seedling emergence. It's also related to the desiccation tolerance that this seed has. That is, if this seed can go through a drying process and still stay alive, and longevity, how long this seed can be stored without losing its qualities. When these four attributes are combined, we have the physiological quality of a seed. So as you can see in this graph on the right, this is a general pattern for physiological seed development. And when these four attributes reach their maximum is when we have the maximum physiological quality. So discussing about seed quality in peanuts, there is some questions that we need answer. For example, when is the optimal harvest time for highest seed quality? Or when are these attributes that are mentioned before acquired in peanuts? Another thing, peanut has dormancy. Can dormancy be a problem? When is it acquired? When is it released? We don't know this answer yet. And because of these questions, we decide to develop the curve of acquisition of physiological quality in peanut seeds. So our hypothesis is that the acquisition of physiological quality attributes will be gradually acquired during seed development, with maximum physiological quality reached in fully mature seeds, or in other words, maximum physiological quality reached when the pod mesocarp is black. Moving to the maturation methods, the experiment was developed at the USDA in Dawson, Georgia, using the cultivar Georgia 06G. The seeding rate was 6 seeds per foot in a field with 6 20-feet rows, 36 inches spaced. The irrigation was weekly, and when we harvest, and after 104 days, we harvest the plants and we process the samples at the Peanut Physiology Lab in Tifton, Georgia. So using the peanut bird profile, we separate the pods into 8 different stages. The stages were white, yellow 1, yellow 2, orange, brown, and black. For the last 2 stages, brown and black, we also divide in 2 sub-stages to identify possible changes within stages. So instead of brown, we'll have brown 1 and brown 2. And instead of black, we have black 1 and black 2. These are my final 8 stages. So the seed tests that I performed were germination, desiccation tolerance tests, and vigor tests. For germination, desiccation tolerance tests, we used 4 replications with 25 seeds for each stage, and the duration of the test was 10 days. The measurements were taken at the last day. For vigor, we used 4 replications with 15 seeds for each stage, and the measurements were taken each 6 hours for 10 consecutive days to evaluate the speed of emergence. And for the 3 tests, we evaluate the radicle emerged with length of at least 2 millimeters. So after separating the pods into stages, we took a sample of these fresh seeds and we separated these seeds into different treatments, because we need to understand about dormancy, when it's acquired and when it's released. So to understand about dormancy, we treated one of these treatments with ethephon, release dormancy. And we performed germination tests for both treatments using a germination paper under controlled environment following the protocol that I said before. We also performed vigor tests for these both treatments using Petri dishes under controlled environment, also following the protocol that I mentioned for vigor. After germination and vigor tests, all the romaine pods were stored in mesh bags, separated by stages, and we placed these mesh bags inside the wagon dryer until reaching 50% moisture. After that, we removed these bags and we placed it in a controlled environment until reach between 6 to 8 percentage of moisture. So we took a sample of these dry seeds and we also separated in 2 different treatments and we performed germination tests on this seed. But it's important to know now that the germination was on dry seeds because we need to evaluate or to understand the response of this seed after being dried. For the statistical analysis, each 25 seeds for desiccation, tolerance test, and germination, and 50 seeds for the vigor test were considered as a replication. The experimental design was randomized complete block, and we ran analysis of variance. When significance was observed, we also ran Fisher's LSD at 5% of probability to separate the means. We used JUMP software to run the statistics and Sigma plot to build in the graphs. Now moving to the results, I will start with the results of germination. So to understand this graph, we have on the y-axis percentage of germination. On the x-axis, we have the stages of development. And we also have the both treatments here, where solid line represents treated seeds and dashed line represents untreated seeds. The first thing that I want to mention in this graph is that after yellow to stage, both treatments has a different response related to germination. Where treated seeds has a high percentage of germination when compared with untreated seeds, which potentially indicates an acquisition of dormancy around yellow to stage. For the percentage of germination on treated seeds, it's possible to observe a high percentage since the orange stage with the highest values between brown 1 and black 2 stages. And for untreated seeds, although there is a variation among the stages, the percentage of germination was relatively stable from yellow to stage until black to stage. In order for dormancy not to be a confounding factor, I will present on the results for treated seeds now for the 2 remaining tests. So for vigor results, I will present the data from orange stage, and we also used 2 indices that can evaluate vigor. The first index is percentage of cumulative germination, where we will have a maximum germination rate. When the maximum germination rate of a stage is higher than 80%, indicate that this stage is more vigor than the other stages. So looking at this graph on the y-axis, we have percentage of cumulative germination. On the x-axis, we have timing hours, and the lines represent the stages. So for this index, all the stages but orange has a maximum germination rate greater than 80%, what indicates that all these stages are more vigorous than orange stage. Also, we can identify that the brown 2 stage was the stage with more vigorous because took less time to reach 80% when compared with the other stages. The second index that I used to evaluate vigor was time required for 50% of seeds to germinate, or T50. The lower the value of the T50, the higher the vigor of my stage. So looking at this graph, we have T15 hours on the y-axis and the stages on the x-axis. For this index, all the stages are significantly equal, but despite they are significantly equal, it's important to know that the brown 2 stage took just 13 hours to reach the T50, while orange stage took almost 20 hours to reach the T50. Moving to the desiccation tolerance results, we have the y-axis, the percentage of germination, and on the x-axis, the stage. Here, it's also possible to observe a high percentage of germination from orange stage 1, with the highest values between brown 1 and black 1 stage. Now, using the data that we have for the 3 attributes, so percentage of germination, T50 for vigor, and percentage of germination on dry seeds for desiccation tolerance, we calculate the percent of maximum value, and then we'll be able to show the physiological quality gain for these seeds. We start with the germination. So for all the graphics we have on the y-axis, the percentage of maximum value, and on the x-axis, we have the stage of development that will represent the seed development. For germination, the maximum values was between brown 1 and black 2 stage. This is the curve that we have for the germination. We did the same thing for vigor, where we have the maximum value at brown 2 stage and obtain this curve for vigor. And for the last attribute, the desiccation tolerance, we obtain the highest values between brown 1 and black 1 stage. And this is the curve for desiccation tolerance. Whenever you got all these curves and put them together, this is what we have for physiological quality gain during seed development. So as my conclusion, based on preliminary results, to understand the maximum physiological quality of a seed, we need to look for the three attributes together. So when we look for the three attributes together, the stage where I have maximum physiological quality, it's brown 2 stage. Because before brown 2 stage, the physiological quality was not entirely reached. And after the brown 2 stage, it's possible to observe that some of these attributes start to decreasing, what decrease the quality of the seed. So this is why the brown 2 stage is the stage with the maximum quality. So with that, the hypothesis was partially rejected because the physiological quality was gradually acquired during seed development. However, the maximum physiological quality was not reached at the black stage, was reached at the brown stage, specifically at the brown 2 stage. So as ongoing and future research, tests to identify the longevity pattern are still in process. These studies being replicated this year, and I also have the same experiment in a different location where I did last year, and I'm replicating this year. And I also have a greenhouse trial last year and this year where I'm tagging flowers daily, collect individual pods to track seed development. All this data together will support to develop the curve for physiological quality in these seeds. So with that, I would like to say thank you to the peanut physiology team, especially my supervisor, Dr. Christian Pilon, the committee members, Dr. Lamb, Dr. Monfort, Dr. Tubbs, and Dr. Gray. I also would like to say thank you to Dr. Chris Butts and Dr. Hans Sorensen from USDA, Zach Cromer, Chris Cromer, and Neil Hobson for the assistance in the farms, National Peanut Research Lab for letting me conduct this trial in their facilities, Georgia Peanut Commission for funding my project, and Georgia Seed Development for donation the seeds. Thank you, and I will make some questions if you may have.
Thank you very much for this presentation, and we do have time for questions, and they've started to come in. The first question is from Stephen Arthur, and the question reads as this: was the moisture content of the seeds measured before subjecting them to germination, dormancy, or desiccation tolerance test? And if yes, what was the moisture content?
So the question was if I take the moisture measurements for the seeds. Yes, I took, and actually after we harvest and we separate the pods, I took the measurements for moistures, and the moisture is relative for this stage, but almost relative, but it's around 18 or 20% of moisture.
Okay. The second question comes from Dr. Dan Anko, and what was the consistent developmental time between each of the pod maturity color categories based on regression using BUs appropriate? Would this limit applicability of these results to this cultivar? And so again, what was the consistent development time between each of the pod maturity color categories based on regression? And would this limit applicability of these results to this cultivar versus other cultivars?
Actually, I collect all the pods at the same time. So with the regression, I was not able to calculate the data between the stages.
Okay. All right. And last question is, we talk so much about the color, the yellow, the orange, the brown, the black, and I would have thought the black would have been more vigorous. What are the physiological differences between the color? We talk about brown and black. What are the differences between a brown seed and a black seed that may account for some of this difference in vigor that you noted?
Yes, actually the question was what is the difference between the colors? Actually is what we are doing, trying to identify if there is a difference, especially between brown and black stage, because we cannot, in my mind, I was thinking that maybe the black stage was the stage with the highest quality. But in this case, we can observe that since the brown stage, we already have this quality. So this is what we're trying to understand. If there is a high difference or if this difference is not so big.
Okay, thank you. And that's the time we have for questions. And thank you very much for your presentation.
Thank you.